The elongation of transcription occurs with a rate of synthesis of about 30 to 100 nucleotides per second. Unlike DNA polymerase, RNA polymerase does not have an associated nuclease activity, and there is an error rate of about 1 in 10 to the 4th to 10 to the 5th. So that means about every 10,000 to 100,000 nucleotides there is a mistake. Of course, this is a mistake in the RNA, not in the DNA, so it is not a heritable change. There's frequent pausing, and this appears to be part of normal movement of RNA polymerase. This may be caused by hairpin formation, so as the new RNA exits from the active site channel, it forms secondary structure. In some cases, this may even cause RNA polymerase to move backwards, and we'll look at that in another slide. And it also helps in the recognition of termination signals, which we'll also look at in a future slide. The transcription bubble is about 17 base pairs in length, and that consists of a DNA-RNA hybrid of about 8 to 9 base pairs. So this is the template strand of the DNA, and this is the transcript. This is the 5' end of the transcript, and this is the 3' end of the transcript. So 8 to 9 base pairs stays in the hybrid position as the new 3' end is being added onto the new RNA. The gene is downstream, so this complex, the RNA polymerase complex, is moving in this direction. The NTPs enter through this channel that's shown, which actually represents a channel on the inside between the beta and beta prime subunits. This is called the active site channel because this is where new nucleotides are added to the 3' end. This is the RNA exit channel where the newly synthesized RNA is released from RNA polymerase, and this is the upstream DNA entering as single-stranded DNA, template DNA, into the channel. The region that is downstream of the active site in beta here and beta prime are called the downstream jaws of the RNA polymerase. As I mentioned in the previous slide, possibly because of hairpin structures that form as the RNA exits RNA polymerase, there is frequent pausing of RNA polymerase as it transcribes new RNA. In some cases, the RNA polymerase even moves backwards, so it's supposed to be moving in this direction, but if it moves backwards, it moves in this direction. And the result is it forces the newly synthesized RNA into the secondary channel. So this is the 3' end of the newly synthesized RNA, which is supposed to be here. Instead, it's been forced here. This is a problem because it stops transcription. Transcription, as you recall, can only add nucleotides to a 3' end, but the 3' end is stuck down here. So this has to be resolved, and that backtrack DNA has to be removed. And this is done by two proteins called GRI-A and GRI-B. GRI-A and GRI-B are able to enter the secondary channel, and the N-terminus of these proteins interacts with RNA polymerase to cleave this RNA down back to a 3' end that is at the site where new nucleotides can be added. So the backtrack DNA is cleaved and removed, and that requires the action of these proteins. It appears that they're not really nucleases themselves, but that in the interaction with RNA polymerase, the complex is able to cleave RNA into short pieces that subsequently then can leave the polymerase complex, allowing transcription to continue with a 3' end where it's supposed to be. Termination of transcription requires two different types of transcription termination signals. One is called factor independent, and that distinguishes it from a factor dependent, and that factor is called rho. Within an operon, there is a termination site only at the end of the last gene or at the end of the operon. So a transcript goes from a promoter through the open reading frames that constitute that operon and terminates after the last gene. Factor independent termination depends on sequences that include an inverted repeat that is GC rich, shown here and here. These are the inverted repeat motifs, followed by a run of A's. The GC hairpin structure forms because of the tight bonding between G's and C's, and this causes RNA polymerase to pause, typically for a long time. So this is the GC-rich hairpin formed by these inverted repeats, and it's shown right here, forming as RNA exits RNA polymerase. In combination with this GC-rich hairpin that causes the pausing of RNA polymerase, there are also these AU bonds between the DNA and the RNA. And these bonds are comparatively weak bonds because they have only 2 hydrogen bonds. So because the AU bonds between DNA and RNA are weak, and the RNA polymerase is paused because of the stem loop structure, RNA polymerase and RNA spontaneously separate, causing RNA polymerase to fall off. So this is the DNA, this is the RNA polymerase, and this is the transcript, which has now terminated as a result both of the GC-rich hairpin loop and the U's that, in bonding with the DNA, formed an unstable structure. Rho-dependent termination depends on both a sequence called RUT and a protein called Rho. So Rho binds to RUT sites, but it doesn't directly inhibit RNA polymerase. So about 100 base pairs downstream of a RUT site is what's called a Rho-sensitive pause site. And it's this pause site that causes RNA polymerase to pause and then causes Rho to unwind the DNA-RNA hybrid. So Rho terminates only if RNA is not being translated by ribosomes. As RNA is made and leaves the RNA polymerase, ribosomes bind and begin to synthesize a polypeptide. As long as the ribosome is moving along the RNA, Rho does not bind. However, if anything blocks the ribosome, this causes Rho to bind to RUT. Rho is an RNA-dependent ATPase, and it also acts as an RNA-DNA helicase. So it uses the energy of ATP, shown here, and unwinds the DNA-RNA hybrid. RNA enters the whole of the RNA enters the whole of the Rho hexamer, and Rho slides along the RNA until it reaches the paused RNA polymerase. So this helicase activity unwinds the hybrid at RNA polymerase. At this point, we don't really know how Rho gains access to the hybrid because we see Rho here, and we see the hybrid is in here. So Rho has to get actually in there in some way to destabilize this complex. So this termination site is what causes RNA polymerase to pause, but it's Rho, which comes in through a RUT site, that destabilizes the RNA-DNA complex, causing the DNA and RNA to disassociate and RNA polymerase to fall off. So Rho-dependent termination is only about 50% efficient, and it appears that factor-independent termination is probably more common than row-dependent termination.